मेरा टाइम है मेरा स्टोरी थी स्टोरी थी मैं भी
अच्छा अब वहाँ पे बोल रहे थे वहाँ पे CBSE वहाँ पे SSC वाला CBSE का दो लेक्चर अरे क्या यार मैं को ऑलरेडी कर रहा हूँ 2 घंटा गया वहाँ के जाना पड़ेगा 3 घंटा 3 घंटा
इसी का है
भाई मां पसंद से वर्षा प्लान सोशल प्लान
सो लोग इधर बैठे उधर बैठे कि गर्मी लगती है हाँ तो स्विच कर लेते हैं डायरेक्शन इज ओवर हियर हाँ
चलो चलो
मेरे को आके बैठ हाँ हाँ निकल से दो ऐसे साइन किया दोनों का ये
अरे यार तुम लोगों की
लग रहा है मेरे लिए उठा भी नहीं मैं डोसा खा के आई हूँ मैं डोसा खा के आई हूँ
ऐसे नहीं होता
तो रोटी में I would rather prefer वो बोरी मिलता है crazy combo see तो बहुत कॉस्ट होता होगा मैं एप्पल के बच्ची यार नॉर्मल ब्रेकफास्ट करने के लिए जाने का है कहाँ जाएंगे 15 मिनट कौन टेबल लगा बड़ा पाव
12 घंटे का ब्रेक है
ऊपर से थोड़ा कुछ
तो फिर और फिर लेट हो जाता है
पहले मुझे
ब्रेकफास्ट करने को नहीं करना
इट इज 40 और सोच आ गई मुझे ना समझ में नहीं आया थोड़ा डिसाइड करके करी तो पीछे मैं अंदर नहीं अरे नहीं मैं लड़ती आप नहीं गुड मॉर्निंग बाय अबे ये तो सोशल से भी है नहीं आना अरे आई डोंट केयर शी अभी नाम मजाक करते हैं अगर मैं बात ये कब है सोशल अभी इसके बाद है 10:45 पे है वो फिर भी ठीक है वो मैं ये तो ड्रॉप करेंगे तो ब्रेक चलेगा मुझे वो नाम कहाँ गिर कर आई है अगर स्कूटी मेरा खेत में तो क्या चली गाड़ी इधर है मेरी गाड़ी इधर है तो ये तो चली कर रही है हाँ चली मैं गाड़ी देखी 200 की यार कितना अच्छा है हाँ वो बहुत नहीं इसने बात किया मैंने बात
कुछ खा के आया
इसने बात किया थैंक्स अच्छा है अच्छा जाते निकाल और बैठ फिर चलना बहुत मोटी
कुछ नहीं मैं तो कुछ खाने नहीं
है वो जैकेट नहीं मिल सकती मैंने ट्रिप पता है क्या वजह है बहुत है लोग एयरपोर्ट बस लोगों को घर पर नहीं है मुझे नहीं पता है तो वही तो मेरी मम्मी
होटल देने के लिए मैं नहीं उठी अपना बेड से। मेरी मम्मी ना खुद से उठ के लेके जा मेरा स्टूडेंट हाँ बट 7:20 वाला बस मैं तो बस में लेती हूँ सेकंड क्लास there's one white color का bus which passes by there was बताया हुआ था बताना मुझे हाँ वो कंपनी में मैं कुछ बोल भी सकती मेरा थोड़ा सा लेट है बस 7:15 वही ना 6:15 प्राइवेट बस है विद्या की बस प्राइवेट से नहीं एक्चुअली सावन से नहीं है ये सावन की जोड़ी अलग अलग है विद्या की बस है
सब आपके नीचे फोन में कोई
चेक करो
नोट है ना शिट क्या
इसका रहेगी इसी this was the main reason I named the school ये सोचे
आपकी बेबी स्कूल में मेरे वो अगर वैसे सोते हैं वो बोलेगी क्लास में नहीं बोला था
हाँ
नहीं ये बोला मैं चाहिए
ये कर पहली हाँ
ये टूट जाता है बहुत जल्दी मैंने कुछ डॉक्टर में सामान किया
आपको टूट जाता है
ये कितना गया वो रहेगा मैं भी नहीं करती हूँ सर मेरा मिनी सुपर
ओके सेम
मिनी सुपर
को मिनी थोड़ा सांगे पसंद है बस
मिनीसो
मिनी सांगे 10 मिनट लेट
there was a reel, okay, and it's like the गाड़ी थी इसमें हेल्प किट बनी हुई थी and उसमें को था this and I'm coming home करके and उसने like किया
बस जोरा का सब
मुझे 150 वेस्ट किया है मुझे जोरा का
काम करेगा जोरा का किसके
150% कैंडी इतना सा नहीं लिया था ना तब मैंने वो जोरा आटे वाला हेलोवीन वाला छी
ऑफिशियल सांगे
मैं तेरे से सीखूंगी पैसे
ऑफिशियल सांगे
तो सारे किया
किया मैंने
वो आइडिया मैंने भी लास्ट कोई याद नहीं आता चॉकलेट दे दी ना
चुप
है ना कोशिश तो बड़ी
मैंने अपना डायग्राम दिखाऊंगी वो वाला नहीं था ऐसा
बड़ो अगली बार दुखी ना
नहीं एक में ऐसी है वो नॉर्मली नहीं
तो वो तो कॉपी है चलो वाली कॉपी तो लाया ना
वो मेरा पढ़ नहीं
तो ये सब तो आता है
वैसे एक्सेल लिस्ट तो दे दो हम तैयार करना पड़ेगा हमारे
प्राचार्य सेक्शन
आई डोंट चलिए जो गलत सामने है
कल मैं वो डेट क्या था
मेरे हाँ वेरी तू कहाँ से हो 15 मिनट में देखो उसके
वो 2 बजे चले जाएंगे हम एक साथ घर जाएंगे ये कहाँ
मैं कहाँ हूँ
अभी तो तू भी चल रही है ना सामने
मेरा क्लास है बाद में
कितने बजे ना भूल रहे थे हम लोग 2 बजे it's a hydraulic. I think my hydraulic is nice. Bhai, मेरा खाने का रोल है. So मैं वो never देखी। ये क्या बड़ा थोड़ा क्यों है? No, what if you said my house? But what are we? मैम को अच्छा लगेगा मैं रात में भी ऐसे दिखाऊंगी
कहाँ लास्ट पेपर फर्स्ट पेपर
सेकंड में देते हैं
पर फिर फर्स्ट
Second.
से कोई है
सेकंड
वो क्या लिखा है I think ये स्कूल में लास्ट बेंच है इसलिए लास्ट बेंच होती है बड़ा है वो ग्रूट है ग्रूट बड़ा है वो ग्रूट सॉरी तो पागल लिखती है तुम अभी क्या उर्दू में बोल रही थी जो बोल रही थी
क्या हुआ ignore at least it's like a hydra
वैसे वो अभी कि उस दिन शेड्यूल था पर कुछ तो हो गया
इतना आगे आगे नहीं बैठ
वैसे ही
नहीं आगे अभी हो गया बेटी अभी हो गया भाई क्या नहीं अच्छा देखो अच्छा तो मैं स्टार्ट ही बहुत so white SSD is छोटे अभी बोलती है आती है पता है मेरी वो ऐसी बोलती है ये सीधा वो और करेगी अभी आई है उधर लास्ट बार की बात करती है एक अभी आई देख आए
Did you label it?
क्यों लिखा
ये what you mean?
अरे नहीं बोला तीन बनाए थे बड़े बड़े
वॉल्यूम से किया
लेट्स सी
एक बार नहीं पढ़ा मैं कल कुछ
पाड़े थे भाई ये जाटी कल को लेट्स के एचआरडी को सिंटैक्स करना लिटरली उसी की जाती है ये हमारे इंडेक्स सॉल्व करना बहुत तो इसको सॉल्यूशन
कुछ नहीं खाने के
हाँ हमेशा ऐसे ही हुआ कि तू बोलती है वैसे हाल अगर चुप आने ना ऐसे करके नीचे रहती है तो रेडी
अरे नहीं आई है तो सो बढ़िया चलती है
They were really rich there. So cool.
अबे घर रोज काम करता है
मैं स्कूल में ये करता
कोई बोला
So now English week up, get ready, with face time.
इसका होमवर्क करा के रखने का मन कर रहा है
But yeah, वो कर रहे हैं.
मैंने 5 दिन कर दिया आपके घर में नाम में
नहीं बात चाहे 10 बजे दिया
15 मिनट अबे वो हाइड्रा है मेरे को हाइड्रा है सॉरी
तुम दोनों अपने टी-शर्ट कटर उठा के
शट अप यार आई एम जस्ट कंफर्टेबल कटर से कहां
वो मेरा बोला पागल ये ले ये वो
कटर से उठा के ना
पागल गटर से ठीक है पहले में
चेरीज
तो अभी टाइप बोल
मैंने नहीं लाया
I wouldn't deny
All
that.
time.
Let's hear. तू भी जाएगा तू जाएगा
नहीं मेरा ये था
ज्यादा feeling है
हाँ,
those
क्या?
are me.
मैं आई नहीं मैं लेता पर देखो भी कर
हाँ सुपर
गर्मी होगी बोला मैंने
ये को सब उधर से ही
तो खेलेगा तुम्हें
मेरी मम्मा
वर्ल्ड कप
तो बस हम मार्क्स इतना नहीं
बोर्ड से ना कभी तो वापस नहीं आना चाहिए
एंडिंग में पूरा सांझी होता है सांझी माय गॉड
वो नहीं आना है
वो किड सेक्शन होता है नहीं फीमेल्स
किड सेक्शन ये वो
फॉरेन में कितने
और खरीदता है वो
बच्चों का सेक्शन होता है दिस इज किड्स नहीं कहते किड्स गर्ल्स
एकदम प्रोग्रेस करते थे वाला सॉन्ग वीडियो
हाँ और मैं अभी मैं बड़ों में नहीं जस्ट सांझी के लिए ऐसे करती हूँ
इससे नहीं आता तुमको पता है जजमेंट ये अभी मंडे आज मंडे आज मंडे
सेम जगह लगा था
अच्छा
वापस साढ़े 6 बजे
अरे
कार्टून से एडिक्टेड
हाँ ये मैं भी मैंने सब कुछ छोड़ दिया
होता है
कौन है वो
चल गया तो मुझे और भी जल्दी फर्स्ट तो चल गया वो बेटा जॉब कर रहा है वो जिम जाता है यार फिर जाने के बाद जिम जाता है बंदा जिम जाता है 400 रुपए आई पर मैं जा रहा था हम 7000 है ना मैं बंदा कोई 20 स्मार्ट मार्क को कोई दुकान है ठीक है वहां से बंदा वेजिटेबल्स बोला नहीं पैसे वेजिटेबल्स 400 रुपए का लाया और वो भी इतना अच्छा है मतलब डिलीवरी
मुझे ऊपर 400
क्या मेरी भी कर दी बोल पैरेंट्स
अच्छा, I have CBSE first. Now, what was the first question we did yesterday? Fragmentation. Okay, let's start with budding. What homework diagrams? You should gonna finish. Diagrams, I'll check which one of you have to show me. First, let us complete with this question. First, let us complete with this question. Diagram, okay, budding and fragmentation, write down. The points, let us write. Budding and fragmentation, right? Budding takes place in unicellular and multicellular organisms. Takes place in unicellular and multicellular organisms. Other side, for fragmentation, takes place in simple multicellular organisms. Takes place in simple multicellular organisms. Other side for bud. Process involves formation of a bud. Process involves formation of a bud. Formation of a bud on the parent body. Which grows to form the complete organism, which grows to form the complete organism, which grows to form the complete organism, remaining attached to the parent body. Remaining attached to the parent body. Other side. For fragmentation, the mature thallus, Thallus, the mature thallus divides to form few fragments. And each fragment complete the growth and form the thallus. Complete the growth and form the thallus. Examples now you write. Examples. What are the examples we had studied for budding? Write one each.
Yeast and Hydra.
And the second write.
Flagella.
Okay, so that was budding, this one fragmentation we had done, budding we had done. And one more is left, that is spore formation that has come across. Vegetative, asexual methods we were studying yesterday. So asexual methods, which are the asexual methods we had come across? Asexual methods we had come across was fragmentation, budding, binary multiple fission. So these were the 4 methods. The 5th method is spore formation. Spore formation takes place in fungus. Spore formation takes place in fungi, the different type of fungus. Now, examples to write first, if I have to mention some examples, it is the mold. What else? The rhizopus. The mold, the rhizopus. Apart from the mold and the rhizopus, all the different type of fungus associated with the fruits also, the different type of mold mucul which grow on the surface of the walls also in the moist weather, and the mushrooms, process of spore formation. So some common examples you should remember. We are talking about the mushrooms, the rhizopus, the mold, etc. Now, how does the method take place? These type of multicellular organisms, they generally have simple multicellular again. They generally have a filamentous body. They will generally have a filamentous body, and this entire Filamentous body is multicellular, simple multicellular organisms these are. Now, in the process of maturation, what will happen? One of these cells, they will start showing multiple nuclear division. So now this process will be something like this, that one of these cells, they enter a process of multiple nuclear division. This entire region is, of course, made up of cells. But here the number of nuclei starts increasing. Something like this that I'm showing you here. So the number of nuclei starts increasing. What is this filamentous thread-like structure known as? This is known as the hypha. Plural, what is it known as? Hyphae. So hypha, hyphae. And then the single cell at the end, one or two cells only will show the process of multiple nuclear division and resulting in a structure something like this. So when the structure is something like this formed, that can be even, uneven also, not necessarily all has to be of the same height. So here, by chance, I'm drawing this way. They can be of irregular lengths, heights also. So this entire knob-like structure where the multiple nuclear division is taking place, this is known as the sporangium. And the filament which is bearing this sporangium, this is known as now the sporangiophore. This is known as the sporangiophore and the sporangium. What is this sporangium consisting of? Large number of nuclei. Now, when this nuclei mature, what now next is, if we try understanding another stage, what will generally happen is this entire sporangium, they will now burst out. This entire sporangium, they will burst out. When will they burst out? When the number of the nuclei has become so many that the membrane cannot hold. A concept quite similar like the multiple fission that we had studied. But there is something extra that we have to study here when we are talking about the concept of spore formation here, rhizopus. So here the entire sporangium will generally burst out something this way. The filamentous structures remain as it is, and all the spores are generally free. So what are these? We will not term this as nuclei, rather we'll have to term them as spores. What are the single cell structure in the fungus which gives rise to the new generation organisms? Spores. Unlike multiple fission, what would happen is each of these spores, they will be generally covered. I now talk about some of these spores. What will happen? These spores, they will be now covered by a protective covering, and this protective covering is known as the cyst wall. This protective covering is known as the cyst wall. What is the meaning? Cyst wall, something like a hard covering of cellulose, as if the pectin, the protein is different, yeah. But just understand it is a hard covering of sugar, as if cellulose, as if, and then making the spore resistant to environmental odds. For example, now that the process of spore formation and the bursting out of the sporangium has happened, probably the weather conditions, temperature, the humidity and the nutrients not very much favorable for the growth of the fungus. So at that time, what is happening? All these spores are going to die out. No, the fungal spores never die out that easily. Why? Because of the presence of the cyst wall, which makes it protect from the environmental conditions, environmental unfavorable conditions. So maybe when this bursting has happened, the temperature was high, the air was dry, so these spores will not be able to germinate and grow again. So within the cyst wall, they can remain active, not dead, but not promoting cell division also even for months. And then again, when the situation is favorable for them, enough of humidity, low temperature, then they will very fast start growing again. So that's the reason we always say that the fungal spores are always there in the air, because they're so small, they're so light, we're not able to see them with the naked eyes. And that's the reason fungal infection spreads very, very fast. Now, this type of examples I'm sure you all must have seen when we are talking about the bread mold. Anybody has seen bread mold? No? Bread mold. Try this. This is the best time to try this experiment at home. Take 1 or 2 slices of bread, keep it outside in the kitchen slab without refrigerating. Just keep it. Now with this weather, now that there's rainfall enough, if you keep it by tomorrow, day after, definitely you'll be able to see on the surface of the bread, some fine cottony growth will be there. Some fine cotton, huh?
Blue color.
It can be bluish, it can be yellowish, it can be blackish. So colors, nothing specific, depending on what species it is. So when we are talking about the bread mold, that is very, very common. Quite a number of times in this monsoon time when we forget to keep the packets of bread inside the fridge, we get to see that.
The keyboard is not working.
Oh, I have put it here. Yes, it's okay. So now when we are talking about the keyboard is here also. Anyways, so now when we are talking about the concept of the bread mold, you'll have to remember that the bread mold that I'm showing you, this is the rhizopus which we generally get to see on the surface of the leather items. Very common nowadays. Now that it is monsoon, even if you take out some of your leather bags and shoes which you have not been using, then you'll get to see on the surface there are some white cottony granular substances as if, and that is the rhizopus. And the mold always on the food items. The mold is always on the food items, on the surface of the bread. Citrus fruits also, any type of cooked item. Maybe some sandwiches are made and etc., and it is not refrigerated. Then also the bread mold in a day or two. So fungus. And mushrooms, all of you are aware. Mushrooms, when we talk about the mushrooms, these also multiply by the way of spore formation. So these 5 methods that we are talking about, this is pure asexual methods. What was common in all of these methods? Only one type of parent involved. There was nothing known as gametes we have been talking about. All the offsprings which are formed are identical to the parents. Variation, we did not talk about any variation, no changes much. The new Spirogyra looks like the parent. The Hydra looks like the parent. I talk about all these fungus and all which will be formed by this method. Huge number they form. So offsprings are in large numbers. We had written already yesterday. Offsprings are in large numbers. So, and all of them are more or less identical. Now we talk about something similar to asexual reproduction, but not actually a method of reproduction, which is known as the concept of spore formation. Let us go to the concept of spore formation. The vegetative reproduction now. We talk about vegetative propagation. We start with this method of cutting. This is layering. We start with cutting. Now, what are the methods of vegetative propagation? 3 methods only we are going to study. Vegetative propagation. What are the 3 methods? The 3 methods would be cutting, layering, grafting. Now this is, we are talking about the next, that is vegetative propagation. What is the meaning? Vegetative for a plant. What is the vegetative part? For a plant, the vegetative part is it's the stem, the leaf, the root, the bud. These are the vegetative part. What is the meaning then? Vegetative meaning, what are you understanding? It is not the reproductive part of a plant at all, but still it can give rise to new plants. It can still give rise to new plants. So talking about the vegetative parts, let us try understanding now what is meant by vegetative propagation. Write down the definition first. What is meant by vegetative propagation? What is meant by vegetative propagation? Which parts of the plant are always involved here? What is meant by vegetative propagation? Let us check. We write down the definition, vegetative propagation, right? The process in which the vegetative parts, process in which the vegetative parts of the plant, process in which the vegetative parts of the plant process in which the vegetative parts of the plant, that is the roots, stem, leaves, buds, etc., give rise to the new plant. Give rise to the new plant is known as vegetative propagation. We talk about the methods: cutting. Number 2, layering. Number 3, grafting. Budding we already discussed yesterday, so we'll not repeat that, but that also is a method of vegetative propagation when we are talking about the plant. Which plant we had discussed yesterday?
Hydra.
Huh?
Bryophyllum.
Bryophyllum. Bryophyllum is a plant which can reproduce by budding. Okay, let us start with this concepts are very, very simple. Cutting. What is cutting? A number of plants, when we take small cuttings of their stem and put them in the soil, in a couple of weeks time, they can establish in the soil, give rise to roots, and a new plant develops. But the method is not that easy when we are talking about the process of cutting. There are some precautions to remember. What are some few precautions? The few precautions are: the stem has to be of a definite length, 3 to 5 centimeters definitely. And the cut that has to be made, oblique. I take this stem, this is the stem of the hibiscus plant. I want to grow another 5 plants here. So what I'm doing is, I'm cutting a stem, this part. And then from this stem, what I'm doing, I'm making small parts again, cutting it this way every time. So I have got now 3-4 centimeter length stem pieces. I'm putting them in the soil, but for 2 months I see nothing grows, because I had been taking the 3-4 centimeter I have followed, but the cut probably was not correct, because I had been constantly cutting them this way. Not supposed to cut it this way, supposed to cut it this way, oblique. But there's a reason why the cut has to be oblique. If at all you have noticed gardeners and all, they would always slash the stem oblique. They will never cut it this way. Can you think what should be the reason? What can be the reason for this? The stem for cutting. You get some case study questions that the stem for cutting has to be something like this. The stem for cutting cannot be like this. This is a wrong way of cutting the stem. It has to be oblique. So if it is oblique and then put in the soil, then yes, very soon we get to see that roots will develop. But with that straight cut, maybe not. Mostly not. What is the reason? If it is obliquely cut, there is enough surface area, because the stem, unlike the leaf, they don't have enough pores on their surface of the body for absorption, water, etc. So, but then the cutout portion of the stem, it is of a bigger area, then the absorption of the water is faster, division of the cell faster, and the formation of the roots will be faster, meaning the upper part of the stem, before it dries out, it should give rise to roots. Otherwise, the entire stem is dying out in a period of 1 month. So 3-4 weeks, enough time when the stem will die out. So before that, if the roots have developed, then it will flourish. So cutting. What about layering? In certain plants, we come across the process of layering. What is it? A plant is there on the surface of the soil. It has got the roots. But at the junction of the root and the stem, quite a number of times we see that this gave rise to some adventitious roots, modifications, and this will run on the surface of the soil, and wherever they are touching the soil, again some roots start developing and a new plant from here. So a number of plants, most common if I talk about the grasses always grow this way. That's the reason if you want to pluck one grass plant, you will get to see the plants in the nearby vicinity also they are getting pulled. Why? Because they are all connected. Why they are all connected? Because they generally grow by this method, layering. So this is one particular method, and this is naturally happening. Jasmine and grapes and grasses, these are common methods which happen where the plant is there. As again I mentioned, some adventitious roots will be developing, some secondary roots will be developing, and they will spread over the soil. And wherever there is a direct contact with the soil, roots develop, new plant coming up. This is the natural concept of layering. Then, where is it? Where is it? This is the natural concept of layering. Does layering happen, or can layering be done artificially? Yes, quite a number of times. Most of the times, now that I talk about some flowering plants, quite a number of fruit plants like the grape, the strawberry, the types of fruits which has got enough commercial demand, and on top of that, they don't have thick, broad stem. Then the process of layering can be practiced. Layering cannot be practiced for mangoes because though it has got commercial demand, but the stem is so broad and rigid. Then what is the requisite for layering? What type of plants can be layered artificially? If the plant has got soft branches, stem not very rigid, then artificially also this method can be done. How? Whenever we talk about these type of plants, and now that they are growing and now they are in the process of giving rise to new stem, new branches, and since the branches are softer, the stem is also comparatively softer, what will be done is these stem, when they are in the process of growth, they will be manually, diverted towards the soil, because the stem is soft. Repeatedly I'm mentioning, they are not hard rigid like the tall big trees. Even when we are talking about this hibiscus and all, these are also comparatively rigid stem. The moment we try bending them to that extent, it will break. So if I talk about the jasmine, softer stem, I talk about the grapes and all, strawberries, softer stem. So now this will be bent this way, but it will not stay here. So that's the reason there has to be a bamboo or a support has to be there. It will be tight here. And a heap of some soil has to be put here. And all this arrangement is done so that the bent portion of the stem remains inside the soil at least for a period of 2 to 3 weeks. Enough for the development of the roots. So now the bamboo or the stick has been attached and then tied with a rope or something. And then this is allowed. And within 2-3 weeks, if all the moist humidity and everything is maintained, then what do we come across? That this particular the stem has started giving rise to roots. And after that, when we pull the stem part, and if it does not easily come out, it is understood that the roots have already established. Then the rope can be untied, detached, the stick can be removed out, the bamboo can be removed out. And we get to see that yes, new plant have started developing now. And then if that is done, then the attachment with the previous plant can be cut down. What is expected now that the new plant grows? Will it be identical to the previous, the parent plant? Will it be identical to the parent plant? Yes or no?
Yes.
Now that this new plant develops, I have removed the bamboo and everything is there, the thread and everything, no need. And now this plant was already developing. So this plant, which is now developing, are we understanding that this should be identical to the parent plant, or it will show new characteristics? Identical, because we have been using only one parent plant to grow these. The third method that we are talking about is grafting. Now, grafting is a very, very important method. Grafting is a very, very important method, and in the entire process of variety improvement, grafting is nowadays done for so many crops. The mangoes that you eat, grafted varieties, the banana mostly hybrids also. Then when we are talking about so many different type of crops and fruits and vegetables, majorly are grafted varieties, hybrid varieties that we can also see. Now, what is the meaning of grafting? What is done? Grafting is generally done to erase, to delete the undesirable characteristics that is present in a plant. What does that mean? This means that whenever we are talking about the concept of grafting, why should a farmer think of grafting? Because that maybe there is one type of a plant. Let me talk about any type of a fruit, maybe now mangoes, I understand. There is one type of a mango plant, and the farmer has seen that the fruits are quite nutritive. The fruits are quite nutritive. The plant resistant to waterlogging. I'm just giving you some basic idea. The process is quite elaborate. The plant, what happened? What is happening there?
Nothing.
Huh? Sit straight. Come this side. Look at her. She's sliding almost. Got up at what time?
Got up at 6.
7 o'clock.
No, 6:15.
Now, plant resistant to waterlogging. Fruits are nutritive. That's a good quality for a plant, good characteristic for a plant. Then plants resistant to waterlogging, meaning if there is more of water in the soil, the roots should not start decomposing. For certain plants, it does. So that's a headache for the farmer. So plant is resistant to waterlogging, but the problem in the plant is, the plant is very much sensitive to pests, meaning the microbes in the soil, water, etc. Other side, there is one more plant, another variety of mango. And in this variety of mango, the farmer gets to see that this characteristic is very good. This breed of the mango is very much resistant to pest. Not a problem at all. But the other side, the fruit may be less nutritive. So for these two type of plants, the farmers would always think of grafting. Because in this, they want to correct this feature. And in this, they want to correct this feature. So they would plan that why not bring the parts of both this plant together, make a hybrid, make a grafted variety, and possible this will get corrected, this will get corrected. And then now they get a type of a crop, they get a type of a fruit with good characteristics. So for that, what they have to do is, the plant which has more of good characteristics, they will keep that attached to the soil. And the plant which is there with few good characteristics, that has to be brought, and a part of the stem, a part of the bud, a part of the root has to be brought here. So something like this. See, the grafting is always done like this, but it is not necessarily root to stem every time. Here I am showing you stem to stem, which is most common method. It can be stem and root, stem and bud also it can be. Root and stem also it can be. And then tie it up. What has to be added here? Enough amount of disinfectants, plant disinfectants are there. Why? Hormones have to be added. Why? Which hormone? Now you should know that. Which hormone should be added here? So that it speeds up the process of cell division. Cytokinin has to be added. Adequate amount of hormone will be added here. Disinfectants have to be added here, because two types of tissues coming together, enough chances of microbial infection. So that has to be handled. And then they quite a number of times they cover it up with wax so that the stems does not get dislocated at all. And after that, in a period of 3-4 weeks time, they will just scrap it off, remove that, or tie with the help of a rope or tapes like this and remain. And that will be kept there for a period of some time. And in a period of a couple of months, 1 or 2 months, it is observed that yes, these ropes and all, they will dry out and fall off, and and now the new plant has started giving rise to the leaf and the fruit and the flower. So now that the two stems have been brought together and a new plant has developed, if a new plant has developed, what should be the characteristics of the new plant? Quite a number of times it is seen that the undesirable characteristic is repeated also. it is not just one stroke success for this type of method. The farmers keep on trying this. Sometimes they are successful. And it is seen that yes, by this method, new varieties of plant is formed. So now what's happening? Now this type, this feature is corrected and they are no more sensitive, rather than this new plant is now resistant to the pest. They are not easily affected by the pests. What was the other problem with this plant? The fruit was not nutritive, and now they come across that in this new grafted variety, the fruit is more or less nutritive like before. So waterlogging also not a problem now. Pests also not a problem now. Fruit also nutritive. So definitely this is a good choice for every farmer. They would always love to grow this. But again, as I say very simply, that we graft it and we get a good variety. It's not just one stroke growing of the plants. They would keep on trying. And then, but is it happening successfully? Yes, quite a number of times. And as I say, quite a number of these fruits, crops, plants that we are growing in our homes, the flowers and the crops and the fruits that we are consuming, most of them today are hybrid or grafted varieties. For example, I'll just give you one or two examples. If I talk about the tomatoes, I talk about the tomatoes. The tomatoes today that we come across, the tomatoes today that we buy have got a harder outer covering. The outer covering is comparatively harder as compared to tomatoes which we probably we were getting almost 20-30 years back, at least 15-20 years back. The outer skin was thinner, easily prone to getting rotten, easily prone to decomposition. So this tomatoes today, without refrigerating also, if you keep it outside, at least 3-4 days they will easily be there. 3 to 5 days they'll easily be there. But talk about the tomatoes almost 15-20 years back, the skin was so thin. It was difficult. So today that's the reason we can buy in bulk and store at home. But if you understand, almost 15-20 years back, 20-25 years back, storing at home was not much of a concept, because the crops used to get rotten so fast. The life of the crop was very short. The shelf life was very short. So buy today, eat and finish by tomorrow. Otherwise it's going to get rotten. But today we buy in bulk almost for the entire week. We don't have time to go shopping every day. But how is it possible? And that's the reason. The demand of the day is we don't have time going for shopping every day. So this entire agricultural concepts also changing, that increase the shelf life of the crop and the fruit so that they can be kept for a number of days. And not only that, exported also. So there are all those concepts to understand. So out of these 3 methods that we studied, what are the 3 methods we studied? Cutting, layering, grafting. Which are the 2 methods which will give rise to identical varieties?
Cutting and layering.
Cutting and layering. And which is the variety, which is the method which will give rise to improved varieties? Grafting. Now in the volume there are some advantages. I want you to read that and tell me if it is understood. Is there any doubt about that concept on grafting, especially advantages of vegetative propagation, that is majorly about grafting mentioned. Please read and please mark that. You get also questions in your paper. Advantages of grafting, especially advantages of vegetative propagation. Now, advantage of vegetative propagation, general, if you are asked to write, what is the general point you write? Plants can be grown in large numbers. We are not dependent on the environmental factors. We are not dependent on the pollination factors. Pollination is just a matter of chance. So we are not dependent on the concept of pollination, fertilization concept, which is a strenuous process, a doubtful process every time for a flower. So that part is not there. We are cutting down the stem of a plant into 10 pieces, and out of those 10, 6 will definitely grow. So we are more or less aware that productivity we are going to get enough. We are not dependent on any agents like the wind and the insect and the water and all these, like we have to when we talk about sexual reproduction. Now the next topic that we are going to understand. And especially for grafting, it is comparatively easier to get improved variety. So please read those points and find out if there's anything that you need clarification on. Done. Read? Anything to clarify? We understood the points? That part is important. Significance of vegetative propagation. You are asked to write. What are the significance of vegetative propagation? Okay. So now next is, show me the diagrams. How many of it completed? This is which one? Okay, the hydra has come alive now here. Don't draw such big diagrams. In a sequence, number one. The diagrams have to be in a sequence. Good. That hydra is so cute. Now draw the tentacles properly. The tentacles should not be all spread out in the body. It's always from one end. Fine. See there, like what she has drawn. But the hydra has to be in stages, no? Only one diagram is there. In your text?
Yeah,
No, for the diagram on budding, it has to be in stages. The diagram that I had shown you here the other day, please take a note. This type of diagrams you are supposed to draw for the hydra in stages. Okay? In stages. The defect in this diagram, the incorrect part in the diagram is the parent and the daughter hydra is of the same. It has to be, daughter has to be a little bit smaller, for that is how you show reproduction. Yes, detached also has to be shown. Hydra, Spirogyra, Bryophyllum. Budding you are asked, fragmentation you are asked, fission you are asked. So these diagrams definitely detached. Detached you have to draw. The parent and the detached offspring has to be together. So always remember for the budding, this type of a diagram. Where is yours? Did you finish? All of you will practice these diagrams. This is okay.
What is the part of tentacles? What are the functions?
Tentacles has got functions like grabbing the food, locomotion also. These tentacles, they can somersault movement that they show with the help bending the body and almost support on the tentacles and again bending and again. That's the type of a movement that this organism show. And capturing the food, holding the food, put it in there in the upper region. They have got a small mouth-like structure. They'll put it inside. So that's the method of feeding also. Helps in all these functions and some defense mechanisms also there. So this stages it has to be drawn. Spirogyra in stages it has to be drawn. Neat, nice diagrams. Each stage generally don't get diagram drawing. And for this is there, and spore formation. Spore formation also, the stages of the diagram has to be practiced. So please mark in your notebook also all these diagrams you'll have to practice properly from this part of the chapter. Cutting, layering, grafting, no diagram drawing there. Now we move on to the next concept, that is the concept of sexual reproduction. First of all, regeneration we haven't done, right? Okay, regeneration is a concept again that is we'll have to look into a concept of regeneration for the for certain organisms. Now, when we are talking about regeneration, now regeneration is actually not a true method of asexual reproduction. Try understanding. When we talk about asexual reproduction, regeneration never is considered as a method of asexual reproduction actually. Then what it is? It is a method by which simple organisms can regain their lost body parts. It is not a standard method of reproduction. Then under what conditions does regeneration take place and how is regeneration? You get this question also. How is regeneration different from fragmentation then? Here also we get to see that the body is getting divided into segments, fragments. No, but here what is the condition? That this particular organism, that is the planaria, if I talk about, this planaria, it may have divided its body into some 2-3 fragments, but not naturally. This has happened accidentally. This has happened accidentally, maybe during predation, or this leaf-like structures, fine, thin body, flatworms, these are. when they come in contact with those hard stones and the rocks of the the seabed and etc. Quite a number of times they enter into the crevices between the rocks and all, and it happens that since they are very soft, their body gets torn. If I again talk about some other organisms like the starfishes, yes, they also show regeneration, and these are also deep sea organisms, and quite a number of times due to predation or coming across other organisms, hard surfaces in the sea bed, what happens? A part of their arms may get detached. But then a part of the arm got detached. This part is not going to give rise to the entire organism. But this part which is detached, this can regain and reform the lost part. Understanding? Is it different from the concept of fragmentation? Yes. Fragmentation, the body naturally divides into few segments. And then depending on the length of the segment, they will give rise to the entire thallus. Here, this is not the concept. Here, the concept is that if this part of the arm is eaten away by a predator, a part of the body detached during movement here or there, then this entire body can regain and form the lost parts. And then what about this part then? They will just decompose. They cannot give rise to the entire body. But regeneration, when we talk about, we also understand regeneration is possible for vertebrates, yes, to a minimal degree. Regeneration, we understand, yes, for the amphibians, for the reptiles also, there is something known as regeneration. Even for us, there is a concept of regeneration for the liver and etc. But that concept of regeneration for us becomes even more negligible. It is not a method where we can understand of ever giving rise to a new organism. But here what is happening, almost like the starfishes and all, what is happening, a portion of the tail is cut, a portion of the limb is cut. A tail may be regenerated, but the limbs and all, they may not be completely regenerated also. Then what is the difference between the regeneration here and the regeneration here? Since it is a very simple organism, and if the body is divided into 2 or 3 parts, since it is a flatworm, it does not have any organ and system, no bones, no cartilage, no complex tissues at all. So this upper part can just undergo simple cell division and form the lower part. The middle part can form by simple cell division the upper and the lower part. And the lower part, if with adequate regenerative cells remaining. Now, if the lower part is this much, this will decompose. This is not capable of undergoing so many cycles of cell division to complete the organism, that is of course. But yes, if the lower part also or the upper part has got adequate number of regenerative cells in the segment, then by simple cell division, they can complete the organism's body. Yes, by that method, we do get from one organism 2, 3 organisms we may get. But how is it possible that 2, 3 organisms they are getting 2, 3 organisms because the middle part can just by cell division form the upper and the lower. The lower part had adequate number of cells, so by cell division they can complete the upper part. Why is it possible here? Why not in a lizard? I cut the lizard into 2 parts and 3 parts. Am I going to get 3 lizards out of those parts? Why not? Because they don't have any well-developed tissues and organs. By cell division, those layers of cells can be formed. Why not for a lizard? Why not for a frog?
Lizards have organs.
Complex tissues and organs are there, and these complex tissues and organs are formed right from the embryonic stage in a vertebrate. Here there is nothing. So in a vertebrate, if I talk about an amphibian, if I talk about a reptile, there is a proper embryonic stage and the growth of the muscles and the bones and the nerves and the circulatory structures has to happen in a definite manner, order. And during the lifetime, when there is a damage to certain parts of the body, can that entire thing be regained back? No, it will not be regained back. Talk in terms of human also, quite a number of times we come across People with amputate this limbs, no, amputation has happened by an accident. They have lost an arm or a finger, etc. During the process of healing, did the bone and the muscle grow and complete that part of the finger or the limb or the leg or the foot? Nothing. Then what has happened? The bone growth has stopped. The muscle growth has stopped. Only the skin, because skin is one part of our body which grows very fast. So skin only has wrapped up the entire region by fast sensory region. Only that has happened. The muscle also will not grow that complete. The bones will never grow. So when we are talking about this, but in case in human also we understand regeneration. What is the concept of regeneration then in human? If no, only that is not regeneration. In case of human, regeneration of the liver, because liver is a tissue in our body, organ in our body, which develops very slow, very slow. So when we are talking about the cell division for one liver cell, quite slower. So whenever a person has gone for some liver transplant or something, surgery or something, it is understood that yes, a small section of the liver, a small part of the liver, can be regained back. But that generally does not happen with the rest of the body, not with the bone, not with the muscle also negligibly. So regeneration and giving rise to new organisms, one giving rise to two organisms, three organisms, in planaria and simple organisms, it is possible. But is this the method by which they reproduce? No, this is not the method by which they reproduce, because they have got other methods of reproduction also. Normally, how do they reproduce? By transverse binary fission. So they have got their own methods of reproduction. Only when they come across an accident, this happens. You get a question in your paper. Is regeneration a method for giving rise to new organisms in complex vertebrates? Why not? What are you supposed to write? What are the points I told you just now? That complex organisms like vertebrates, they have got tissues,
complex
bones,
tissues
muscles,
which cannot be regenerated
at any time during the lifetime. It is grown in a pattern, a proper pattern, right from the embryonic stages. Okay. So that concept we'll have to write. Let us just complete one or two questions and we move on to the next segment of the chapter, that is sexual reproduction. Look into the previous board questions. Look into the previous board questions. Number 4. Number 3, number 4. See number 7. This type of a question can come in your paper. Number 7. Now, if you have not drawn, you can draw the diagram drawing. If you want, you can draw it later at home also. Number 25. Number 25. Number 20. See, yesterday we had done this.
Number 20.
Number 20. A and B. Number 20. A and B. And tick number 26 also, that we will discuss. You can write down the answer there in the textbook. Come on, all of you will write down all these answers properly in the notebook. Fast. Then we'll have to start with the next one, that is the reproduction, sexual reproduction in the flowers first, in the plants. Come on, start your work, all of you. Wherever there's a doubt, you clarify. Go through the questions, all of you. Find out, you know the answers, you don't know, there's a confusion, please clarify and write. Don't take too long, 10 minutes should be enough.
What is the meaning of what happens when a natural paragraph limit attains considerable length?
It divides to form specific fragments. It divides to form specific number of fragments.
Diagram we need to first
Diagram is about which one?
East.
East. Keep space, draw later. Diagrams, the way diagrams you all are drawing, you will not get full 2 marks. Diagrams has to be neat, nice, perfect drawing. Then there's no need, there is no reason why you should lose mark for the diagram. Simple diagrams you can easily practice. Now in this next part of the chapter also there are diagrams to draw. In the first part of the chapter, now that we are almost coming to the close of winding up with the syllabus, in the first chapter, life processes. What are the diagrams?
Differentiate between binary fission amoeba and binary fission leishmania.
Read the question.
Differentiate between binary fission and amoeba and binary fission and leishmania.
Leishmania, the binary fission is transverse. Okay, I'll give you that answer. Once you reach there, tell me, I'll give you that answer. Leishmania, they show transverse binary fission.
And how does diffraction take place in? Okay, never mind.
That we have that. Plasmodium. I'll show you that figure so that.
We also did not do the diagram for brain.
For brain, we haven't done.
And also like reflex action, that arc.
Reflex arc.
Yeah, like motor and neuron and
That diagram we haven't done. The components I think we have drawn, no?
Yes. We in a flow chart.
We did not
Okay.
do
The diagram. Okay. brain diagram. Okay, this few diagrams I think we are left with. Let me complete this part of the chapter, then we will do. What am I trying to show you? Transverse binary fission for Leishmania. Come on, write down that answer. Okay, let us write down that answer then. Binary fission in amoeba. Binary fission in Leishmania. Okay, write down. Number 1, the first point for binary fission in amoeba. The process takes place in Leishmania is also a pathogenic parasite. It is.
What disease does it cause?
This causes when we are talking about this Leishmania, it causes sleeping sickness.
Oh,
Sleeping. No, insomnia is not sleeping sickness. It is not insomnia. Sleeping sickness is a condition where this parasite starts infecting the nervous tissues, and the infected person will always feel drowsy. Apart from that, there are other effects also in the body. But insomnia is not an infection. Insomnia is to do with the stress levels and the improper coordination of the brain, nerves. Takes place in, for amoeba, you write, this process takes place in free living unicellular organisms. Free living unicellular organisms. For Leishmania, takes place in pathogenic unicellular organisms. Pathogenic unicellular organisms. Number 2. Nuclear and cytoplasmic for amoeba. Nuclear and cytoplasmic division. Nuclear and cytoplasmic division takes place once dividing the parent cell from the central region. Dividing the parent cell from the central region, and the constriction moves towards the center. From the dividing the parent cell from the periphery. Periphery means the outer region. Just correct that, all of you. Dividing the cell from the periphery towards the central region. Periphery towards the central region. Other side for Leishmania.
And the constriction, like periphery.
Complete the sentence. Dividing the parent cell from the periphery, and the constriction deepens towards the center. Constriction deepens, D-E-E-P-E-N-S, deepens towards the center. Other side, for trypanosoma, the parent cell divides. See here, what we have written is that when we are talking about the amoeba, from the periphery and deepening towards the center, and this entire process is horizontal. Horizontally it is taking place. So within bracket you can mention also or comma horizontal. So it is absolutely horizontal method. Trypanosoma I am showing you also parallelly. It is vertical method. Okay. Now, next side, the other side. Parent cell divides in such a way in such a way in which the cytoplasm and the nucleus divides vertically from the periphery towards the center. Okay. So this is the difference in trypanosoma. Go to the other question. There is one more question on the east that you'll have to mark the parts. That you can do in your volume itself. Mark the parts. The parts are 1. Can you see 1 there? Cytoplasm of the daughter yeast. Cytoplasm of daughter yeast. 2. Young bud. 3. Cell wall of daughter yeast. 4. Cell wall of parent yeast.
Ma'am, what is second?
Second, young bud. It's a young bud showing there. Cell wall of parent yeast. Go to the next. Check what else I gave you now. Check, read that question.
A student tried to observe the slide of binary fission in amoeba under a microscope. After focusing on the slide, he was not able to get a clear view, even after adjusting the diaphragm. Suggest 2 possible modes of adjustment of the microscope to get a clear view of the slide.
Now, when we are talking about the placements, now this type of a question, the question is that when we talk about this type of a microscope, let me just show you a simple one. All of you must have seen this microscope, right? Now, when we are talking about this microscope, where he must have put his object here. This is known as the stage of the microscope. So can you see the slide also seen here, the glass slide? So on the glass slide, he must have kept it. But when he tries to see, he is not able to see anything. That is what is mentioned there. He is not able to see anything. What type of adjustments he has to do? Number 1, he has to place the object exactly below the lens. And wait, I'll give you. He has to put the object exactly below the lens. The focus of the lens should be exactly on the object. And the mirror has to be adjusted so that enough light falls on the object. That is number one. Then we get to see a clear view. Maybe not the object very clear even then. So that's the first thing that we'll have to check. And then second what? Second, I can see the field now, but still I can see the dots are there, but I'm still not very clear about the object. What I will do? I will have to adjust with the major and the minor screws. These are known as the major and the minor adjustments. So the major and the minor adjustments has to be done, and then I'll be able to get a clear picture. Did you ever see anything on the microscope?
Yeah, I had seen the same thing. I couldn't see it, like amoeba, the same.
Wait, you could not see in the school? No. The first thing is, with the help of the major adjustments, the focus has to be corrected, and then the mirror, the light. That part is the first one. Second, still we are not able to see, then with the help of the minor adjustments, we'll have to keep rotating and rotating unless and until we are able, and we will definitely be able to see. The amoeba and the paramecium and then all these structures are so beautifully seen under the microscope this way. Okay, so let us write that answer. The following adjustments have to be done. The following adjustments have to be done. Point number one. Following adjustments have to be done. I'll take a oral next period for the last 5 minutes. Last 5-10 minutes. You are comfortable with which chapter? Life process is all comfortable now?
Yes.
Yes.
Okay? Last 10 minutes. Because otherwise we don't have separate time for revision. Now that we are almost winding up with the syllabus, we will have to do a little bit of it. And there will be one class only on diagrams that I will take. I'll try giving you a worksheet also on that. And we will do a class on diagrams. Okay, continue this. The following adjustments have to be done. Number one. The object should be placed exactly below the lens. By adjusting the slide on the stage, comma, using the major adjustment screws, and adjusting the mirror so that enough light falls on the object. Adjusting the mirror so that enough light falls on the object. Point number two. The fineness of the object will be obtained, or the clarity, the fineness of the object will be obtained by the adjustment of the minor adjustment screws. The minor adjustment screws. So the bigger one is known as the major adjustment, the smaller one known as the minor adjustment. Okay, so now we'll have to take a break. After the break again, take a break. After the break, we continue with the sexual reproduction in plants.
Healing and
नेक्स्ट
अभी
अरे मतलब इतना भी बकवास नहीं है, थोड़ा बहुत खा सकते हो, मतलब ऐसा नहीं है। अब वो
हम बाहर से बुक लेकर कहाँ से मुझको फोन लेने दे मेरा
वो वही लाने जा रहा है
नहीं,
नहीं आटा के पागल तू बदल
तो फोन ले गया तो मैं ना
क्या मिली क्विट लेकिन
अगर मेरा बड़ा पाव खा के हुआ तो
एक चुप सुबह सुबह बड़ा
भाई अम्ब्रेला लेके जाओ मेरे को भूख लग रहा है अम्ब्रेला बारिश हो रही है
आज ही लेके चली जाओ
सीरियसली पर लोगों ने लिया अरे जीव ना शट अप
उनको ने भेज दिया तो जाओ तुम मैं जाऊंगी
मेरे को चाहिए बनाने को अभी बहुत
इधर है बिल्कुल याद नहीं
अरे नहीं डिलीट कर सकते हो
भाई तो वो छोड़ ये कर ले ये कुछ ही नहीं
नहीं वो लास्ट स्टेज में वो जरूरी हाँ
चलो ठीक है